the steps. Now, before I say that, I just want to remind you that we're looking for a particle that's very small, that has a protein coat and some genetic material, either DNA or RNA. So the last thing you want to do is mix it with other things with DNA or RNA or protein, because you might get confused then. So what did Enders do, and has been done in every isolation experiment for the last 63 years? Here's what they did. They took the mucus from somebody with measles, and they washed it with some saline, and then they put it through a filter paper like you would make coffee, right? And so the bacteria and the fungus and the dead cells, they get caught, and you get this liquid of anything dissolved in the lung fluid. Now, in there are proteins, toxins, water, viruses, and probably a whole lot of other things. So that is not, nobody would say that's purified, isolated virus. Then they mixed it with milk, or now we mix it with fetal bovine serum, which are sources of proteins and genetic material. And then they inoculate that, which means they spread it on monkey kidney cells, which are another source of proteins and genetic material. Sometimes they mix it with amniotic fluid from a cow, or fetal bovine serum, or horse serum, or other things. Now, nothing happens then. So then they have to take away the nutrients from the culture. That's called minimal nutrient medium, and nothing happens. So then they add antibiotics. Now they add gentamicin, penicillin, streptomycin, and an antibiotic called amphotericin, all of which are toxic to the kidneys. That's what it says in the PDR. And they're spreading this on monkey kidney cells, and then lo and behold, the monkey kidney cells break down into millions of different little pieces of protein and genetic material, and they say that is the virus. Now, think about that for a minute. How do you know that this came from the original person with measles and not from the breakdown of the monkey kidney cells?
Yeah.
Well, interestingly, John Enders did a control experiment of sorts. He did the whole thing, except he started with nothing from anybody with measles, right? So he put the bovine fetal serum, the horse serum, the embryonic fluid, he spread that on monkey kidney cells, he took away the nutrients, he added antibiotics, and it broke down, and he said, quote, "It broke down in a way that is indistinguishable from the first culture." Now, let that sit in for a minute, because if you're a scientist, what you think is, well, this had nothing to do with the measles virus, right? The stuff from the measles. This has everything to do with the fact that you're starving and poisoning tissue, which inevitably breaks down into these little particles. And somehow his warning that someday somebody's going to have to figure out whether these, quote, particles are from the measles person or from the kidney cells. And literally for 63 years, nobody until last month repeated the experiment. Now they just say, here is